How do plants know where and how to grow? We're going to look at plant hormones in this lecture. In this lecture, be able to define hormone, tropism, that includes gravitropism and phototropism, auxin, cytokinin, gibberellin, abscisic acid, ethylene, the triple response, and thigmomorphogenesis. Give a mechanism of action for each one of the plant hormones we covered. Compare hormone deficiency, which is an inability to make the hormone, to hormone insensitivity, which is the inability to acknowledge the hormone's existence, and be able to identify plant responses to stimuli, such as light, gravity, touch, flooding, heat, and more. Plants receive signals in one part of the body and need to respond in another part of the body. When this happens, the plant must send a signal from the part that receives to the part that must respond. And this signal is often a hormone. A hormone is a small chemical messenger produced in one part of the organism and received in another. In class, we're going to look over some examples of hormones in humans, which we will be covering in later lectures. Plants do move. Although we often think of plants as somewhat static, they do very slowly move, and if you speed up a camera, you can actually see them bending towards things. A tropism is the growth toward or away from a certain stimulus. It is notable that tropisms are directional. This experiment by Darwin was done to show how plants bend in response to light. What you can see is this is a phototropism, where the cells on the illuminated side actually grow slower and smaller than the cells on the shaded side. While this may seem counterintuitive, it means the cells on the shaded side will be longer and cause the plant to grow towards the light. Gravitropism is a similar thing being done in the roots, that is in response to gravity. We'll see some little pictures of that closer to the end of this lecture. So let's cover different plant hormones. We're going to look at 5 hormone classes: the auxins, which are responsible for plant tropisms and adventitious roots; the cytokinins, which increase cell division; the gibberellic acid group, which generally act on the internodes and on germination; abscisic acid, which governs some stress responses; and ethylene, which governs something called etiolation and also cell death. First up, these auxins. Auxin is produced at the apical meristems. That would be the root apical meristem and shoot apical meristem. It is transported to cells that are elongating. This causes a selective elongation, which makes a stem or root bend. And I'm going to draw this on the board, but it's like the figure we looked at a little while ago, where you see the cells on the shaded side or the cells on the side away from gravity are going to be longer. So how does a plant make longer cells? Well, that is the acid growth hypothesis. Auxin increases the activity of proton pumps, which put protons, these little H+ ions, into the cell wall. Expansins and cell wall loosening enzymes are going to cause the microfibrils in the cell wall to be broken down, allowing for the cell to elongate as the cell takes up more water. That pressure is going to push the cell membrane against the cell wall, and where these polysaccharides have been digested in the cell wall can expand, thus elongating the cell in a directional manner. Auxin is also used to create adventitious roots. If you were to make a wound in a plant stem and then apply some auxin to this wound, the stem will begin producing roots. That's an auxin powder you can actually buy. This can allow you to cut a stem and then put auxin powder on it, and the stem will begin making roots. You have to wrap up the stem or it will dry out, of course. And this is allowing something called plant theft. So if you're ever walking through a fancy garden, you may see someone snip off a small stem and jam it in their pocket. They're going to take it home, put it in auxin, and it will develop roots. And after it develops roots, they can put it in a pot, and then they have a copy of the plant. Cytokinins are another hormone that induce cytokinesis. So cytokinesis is the division of the cytoplasm after mitosis. So yes, these are mitosis-causing enzymes. Cytokinin and auxin work together to stimulate growth. Auxin is released by the apical bud, and the presence of auxin is going to inhibit the growth of axillary buds. And inhibiting the growth of axillary buds is going to make sure that, well, the plant's not going to be branching right next to the apical meristem. However, farther away from the apical meristem, you'll have the plant beginning to branch as the auxin to cytokinin gradient changes. If the cytokinins dominate over the auxins, you'll have the growth of axillary buds, which would happen if you removed the apical meristem. So this is called apical dominance, when the apical meristem causes the inhibition of more branches. If you want a plant to become branchier, you simply snip off the apical meristem, thus removing auxin and allowing cytokinin to do its thing. Cytokinin is also used to increase cell growth in callus formation. So plants have the unique ability to make a new plant from any plant cell given the right conditions. This is called totipotency. And the cytokinin to auxin ratio governs whether you get a shooty callus with a bunch of shoots or a rooty callus with a bunch of roots, or just increase the size of the callus. So growing these little plantlets on a medium with plenty of auxin and cytokinin can change how the plant grows and can allow you to clone these plants. This is useful in genetic engineering, where you want to take chunks of plant, genetically engineer them, and then grow whole new plants from them. The gibberellins are a group of plant hormones that are largely responsible for internode elongation and germination. So what is internode elongation? Well, you may remember from us looking at a stem that there are spaces between where the leaves come out. And gibberellins are responsible for making those spaces longer, which causes a plant to be taller. This can be seen in a process called bolting, where you have a plant like this Arabidopsis plant that is in rosette form with its very short internodes, thus putting its leaves very close to one another. When it comes time to grow seeds, though, it doesn't help to have all the flowers and seeds right next to the stem, so it increases the internode length and makes this long stem that can, well, grow up high and bolt. It's helpful to spray gibberellin on grapes because this allows more space between the grapes, thus giving the grapes more room to grow and increasing the total value of the grapes as there are, well, bigger grapes. Gibberellins also induce germination. They start the germination and allow the root and shoot to come out of the seed. Spraying certain species of plants with gibberellic acid actually causes the seeds to germinate. This is a little gardener's trick if you're trying to grow things indoors and it's not the right season. Just spray them with gibberellic acid and some of them will germinate. Another trick is you can actually use liquid smoke to cause other ones to germinate. And this hints that there are other conditions at work here, and there are other hormones at work. So once again, let's look at some more hormones, like abscisic acid, which is also involved in seed dormancy. So abscisic acid will cause the seed to be dormant. So you may think abscisic acid and gibberellic acid are kind of going against one another, and you'd be pretty much right. Mutants in abscisic acid will germinate early. Plants that underproduce abscisic acid will have early germination of seeds, which can occasionally happen in the fruit or on the cob if you're looking at corn. This can be beneficial, though, for things like the red mangrove, where you get this early root coming out, and this early root coming out allows the mangrove to hit the soil running, as it were. When it's dropped from the adult, it'll be able to plant itself because of that extra long root. So underproduction root mutants do not make enough abscisic acid, and reception mutants can also just not see the abscisic acid and thus not have any seed dormancy. Ethylene is another plant hormone that is responsible for behaviors of the plant during germination. If a plant is kept in the dark, it will start having these little behaviors where it elongates a whole bunch and the stem becomes very thin and it grows straight up. This allows it to find the light. But what if the plant hits a solid object? Well, it slows the elongation, thickens the stem, and then begins to curve. As it curves, it'll go around the object, and the thickening the stem can allow it to even sometimes move the object. Ethylene mutants are insensitive to ethylene. So you see this ein mutant, it is unable to receive the ethylene response that causes the triple response, and thus it elongates even when ethylene is being present. The CTR mutant is a constitutive response, and that is going to overproduce ethylene and have the triple response even in the absence of ethylene or the absence of a heavy object. Ethylene is also responsible for cell death, and while that may sound like a bad thing, you must remember that cell death is a normal part of organismal development. This is why your fingers aren't webbed, because the cells that would be between your fingers have died. For plants, this is responsible for things like seasonal responses, where the abscission or cell death of the space between a leaf and the stem allows the leaf to come off of the stem during the fall. Ethylene is also responsible for the cell death during fruit ripening, which is why you can actually grab green tomatoes, gas them with ethylene, and get red tomatoes. They don't taste like the vine-ripened ones. Plants respond to other stimuli. Here's an example of gravitropism, which I mentioned earlier, that the root will bend towards the gravity as these statolysts, which are little starch granules, fall to the bottom of the cell. This actually tells the cell what direction gravity is acting in. It's kind of cool. There's thigmomorphogenesis, which is the response to touch. And if you touch a plant too much, it'll grow smaller. This helps it. If it was being touched too much, it may fall over under all the touch. So it's helpful to be smaller if you're being constantly brushed against by other plants. There are stress responses as well. For example, a plant under drought will make more abscisic acid, which immediately causes the stomata to close. Over the long term, a plant may wilt, thus exposing less tissue to sunlight and thus not drying out as fast, or selectively lose certain leaves, which also reduces the amount of water that the plant needs and allows it to survive in drought longer. The opposite of that would be flooding, where the plant now has a whole bunch of roots underwater, and underwater there's not much oxygen available. Remember, roots need oxygen. So this allows an ethylene response to cause aerenchyma, or air tissue. This causes selective death of certain cells, that's what ethylene does, making open spaces where air can actually get down to the roots. Heat stress is similar to a drought stress because heat and drought often coincide, and this is also heat shock proteins, which allow other proteins to not denature under the heat. A cold stress changes the membrane lipid composition to have more unsaturated fatty acids. Plants can also produce antifreeze sugars, which prevents the cell water from freezing. In animals, we'll see antifreeze proteins to prevent the cells from freezing.